Thank you very much, Dr. Tulahouse. It's a pleasure for me to speak to you here from London, which is a sunny day, but still very cold. And I thank Dr. Tulahouse also for inviting me to speak to you this morning for you and this afternoon for me. This is an area which I've been working in, particularly looking at the effect of pollutants, and it's an area where we are now very gradually looking into how pollutants and irritants and allergens affect the or interact with the epithelium. These are my declaration of interest. I've already sent that previously. Right. So the airway epithelium is at the interface of the environment and the lung or airway tissue, and obviously the epithelium is exposed to all those things that are carried out in the air that we inhale, and this is not necessarily an exhaustive list of things that the air or what's contained in the air that we breathe, and obviously we can get exposed to cigarette smoke, plant and insects, house dust mite, diesel exhaust, the pollution that we get exposed to most commonly, those of us living in cities, but also other things that we use at home, chemicals, detergents, etc. And obviously, more importantly, as we get into this climate change thing, is I think about those pathogens that we inhale, including bacteria, viruses, and fungi. And of course, there's been a lot about microplastics, a discovery of those micro or nanoplastics that can inhale and penetrate deep into the lungs, which is quite a new part of the exposome. And the other actor, of course, on the scene is the epithelium, because this is the first layer of cell that's going to be in contact with those insults, those parts of the exposome that we're all exposed to. And in fact, this slide really summarizes what I'm going to talk about, and about the epithelial response to those insults. And the thing that's becoming very much established now is that those insults does change the permeability of the epithelium, such it allows those insults to penetrate more deeply into the tissue, into the airway tissue or in lung tissue. It goes down deep into the alveolar epithelium. But the interaction of those insults with the epithelium also activates the epithelium in various ways, and inducing inflammation. And a very important part of the inflammation is the so-called type 2 inflammation, which in fact forms the basis of asthma, and also the activation of other inflammatory pathways, particularly Th1, Th17, etc., which form the basis of the inflammation that we can see in non-asthmatic inflammatory conditions such as COPD. So, so this is this slide is basically a resume of the previous slide, of course, but showing a bit more the sort of disruption that we get at the level of the epithelium that leads to epithelial shedding. And this is the interaction, the effect on the various proteins that keep cells together and prevents leakage, and in asthma particularly, there is evidence that there's a impairment at the level of gap junctions, adherence junction, and tight junction, as listed here. And that thickened basement membrane that we see in asthma, in fact, is not really a protective layer. The major protective protection is those junctional proteins here that are affected, we know, in asthma, but also affected by those elements of the exposome, particulate matter, respiratory viruses, and allergens. And down, and I've already mentioned this, the inflammatory response also, the downstream inflammatory events. And here, particularly emphasized is the secretion of those, what we call alarmins, IL-25, IL-33, and TSLP. I've already mentioned this, but I just want to mention that asthmatics are particularly much more prone to the effects of those external insults. And one of the reasons is that the cell types that we see in asthma is quite different from normals. And this is some work that was done by Professor Nouwen in Groningen, using single cell sequencing, comparing control healthy epithelial cells, airway epithelial cells to asthmatic airway epithelial cells, and noting an increase in the epithelium of asthmatics, activated and cycling basal cells, increase in mucociliated cells and goblet cells, and this we already knew that, and also an increase in T cells and mast cells. So that the environment is probably conducive to a much bigger interaction of the asthmatic epithelium with those external insults. The presence of non-adherent epithelial cells in severe asthma, I just want to present some evidence that we are gathering at the moment in a study that we're doing in patients with severe asthma, particularly. And we've been involved in a study where we've been collecting nasal epithelial cells from patients with severe asthma and culturing them. And we found that in a large proportion of those patients, when you culture those nasal epithelial cells, they don't adhere, and we don't get a good culture going on, contrasting to those controls or non-asthmatic subjects where you get a very nice adherent cells that are proliferating, and of course we also see that in some patients with severe asthma, and these are the and when we then measured the resistance, the electrical resistance of those cultured cells to as a measure of the barrier integrity, we see that those that have shed epithelium had a lower resistance than a non-shed one. So the non-shed one obviously have a much more continuous non-leaky epithelium. And the difference between those non-shed and shed epithelium is one of an increase in eosinophils compared in the shed compared to the non-shed. And so that just implies that there may be the influence of the presence of eosinophils, perhaps through the release of eosinophil proteins that causes this this leakness and an inability to get a culture of adherent cells when we culture them. And this is ongoing work. And we're just working on the mechanisms by which the eosinophils is causing this shedding. Of course, we also see abnormalities of the barrier function in COPD, and this is really just to emphasize the fact that maybe this is likely to be secondary to a neutrophilic inflammation, and also through a a sort of neutrophilic response in terms of the interaction between cigarette smoke, which is one of the major cause of COPD, and viral infection and so on, that induces the production of those neutrophilic type cytokines or chemokines to recruit neutrophils, and also some element of activation of those T cells, and there's evidence that there is abnormalities of those junctional proteins, although those abnormalities are a bit slightly different in terms of when compared to those that have been described in asthma. Now, just a few examples of some of the chemicals that can increase epithelial permeability, and most of those studies that are being done now are studies that use air-liquid interface cultures, and this is a constituent of flame retardant, polybrominated diphenyl esters, and there's been some issue about the toxicity. And this study, an Italian study, demonstrates that these diphenyl esters does cause an increase in epithelial permeability as measured by transepithelial electrical resistance measurement, as shown here. And also you can see that there is it affects the Z01, one of those junctional proteins shown here on immunofluorescence. And also you can see how the epithelium is getting detached with those diphenyl esters. There's also been similar data produced looking at detergents. And in this study, what was interesting was that it was also done not only on epithelial cells obtained from healthy, but also from asthmatics and from COPD, where you could see also this change in transepithelial resistance, and also a greater effect that's been shown here in those epithelial cells obtained from COPD patients. And these are the results of the paracellular flux, and also the immunofluorescence of Z01 and also of occludin, which you can see being affected here. But what is interesting in this study is that there's been some analysis of the potential mechanisms associated with this disruption of the tight junction barrier, and analysis of those transcripts here of those cells indicates, summarized here, that there's an impaired tight junction integrity, although the transcript amount is not changed, but the integrity of this is changed. There's increased lipid metabolism and of apoptotic genes, and also there's increased expression of IL-33 and TSLP, with all those alarmins produced by the epithelial cells, a reduction of cell adhesion genes, and a damaged cell membrane lipid, and that might explain this this disruption of the tight junction barrier. And in addition, this is the inflammation that's associated with type 2 immunity, and you might expect that those detergents may induce type 2 immunity, and perhaps may cause an asthmatic inflammation. Another aspect, now coming down to the more common type insult on the respiratory, on the airway epithelium, is of course PM2.5. And this study, I think, is important because it shows that the organic extracts, which is a very important component of PM2.5, that much more active than watery extracts, as they've shown in this study. But these are the effects of the organic extracts. And two things I've shown here, which is the effect on MUC5AC positive cells. And you can see, I mean, these are mainly goblet cells. So to high levels of this organic extract of PM2.5, you can see here. And this is contrasted with when those epithelial cells are exposed to IL-13, which is well known to induce this goblet cell hyperplasia and an increase in MUC5AC. And also there's an increase in secretion of the mucin 5AC. The other aspect that's been shown here in this study is the effect on FOXJ1, which is an important component of ciliary of ciliated cells. In fact, it's very important for the ciliary cell activity, and you can see that there's a very much reduction in the expression of this FOXJ1, shown here, high OE, and also a much bigger effect shown here than the effect of IL-13. And so that might explain why there's been various studies that have shown that the ciliary beat frequency is affected by PM2.5, and the PM2.5 extracts also obviously contain those PAHs contained in PM2.5. Now, in the same study, we get also some idea of the mechanism by looking at the differentially expressed genes that you see on the addition of PM2.5 organic extract. And you have these genes, the functional effects are shown here in 3 classes. The aryl hydrocarbon receptor response battery, that's got to do with the metabolism of certain things, also about CYP1A1, the inflammatory mediators, particularly IL-1A, IL-1A and IL-1 beta, IL-1 alpha and IL-1 beta, and the airway mucus program, which of course we've seen the MUC5AC response, and that's also reflected in those in this figure here, upregulated differentially expressed genes in the mucous secretory and ciliated cells, and also the various pathways that are being sort of activated by PM2.5 organic, which I won't go into details, but as you might expect, the cytokine, the chemokine signaling, and the cytokine activity, and some evidence for MAP kinase signaling pathway. Now, what about the effects on various oxidative stress substances, and I think we all know that these pollutants, PM here in PM4, not PM2.5, are showing here first the reduction in the total oxidant capacity of those alley cultures of human bronchial epithelial cells, either exposed singly, one exposure, or exposed repeatedly on 3 occasions, and here on 3 occasions, as you might expect, a bigger effect in a reduction in total oxidant capacity, and that's associated with an increase in those oxidants, evidence of increase in oxidant production, malonaldehyde, 8-hydroxy-DG, and a reduction in superoxide dismutase. So, so a reduction in antioxidant and increase in oxidant ratio and also reflected very much in the DSSG, and associated with the production of those cytokines, as shown here. Now, this is from a very nice French study, and 2 years later, they published a more comprehensive study that looked at compared low PM dose to high PM dose, acute exposure to repeated exposures, as they've done in that first study, and of course, what was interesting here, of course, they also looked at normal human bronchial epithelial cells, but also cells from COPD patients. And I think this is very much an accepted paradigm of the response. So when you're using low amount of PM, low amount of pollutant, and exposed ones, this induces a pretty low level of high ROS, of reactive oxygen species, compared to if you have repeated exposures or high level of PM 2.5 exposures, you have a high levels of ROS. With low ROS, the cellular system, if I may say so, or the cellular defense works very well. So you activate NRF2, which activates NRF2 target genes, and that can control the amount of reactive oxygen species, so you get regulation, and so you don't get oxidative stress, and the mitochondrial function is preserved, and there's no inflammation. While contrast to if you have a high amount of PM 2.5 exposure and you have repeated exposure, the systems, the defensive system is overwhelmed, and you get oxidative stress, and that has an effect on mitochondrial function, and that has a result on increasing inflammation, and also on when it gets in extremis, on cell survival, and there's an increase in cytokine production also. So I think this is generally an accepted paradigm that's used, and obviously that can be applied to the human situation. And I guess if you are exposed to a lot of pollution, your system will get overwhelmed, and that might lead to the medical consequences of pollution that we know well. This is a very complicated slide that we had in a review, published in a review a few years ago. But I think focusing on the importance of this oxidative stress, and really summarizes some of the mechanism by which oxidative stress causes mitochondrial damage, activates or interacts with the DNA and causes damage, and induces inflammatory response. I'm not going to go into this in much detail, and also effect on protein folding, and that either inactivates certain proteins or activates certain proteins, depending on the context. Right, thank you. Let me go to the next one. And so, and that leads me to whether we can have this concept that the exposure to air pollution leads to chronic lung disease. And I think this has always been something that over the last few years, and I think there's at least epidemiological evidence for some of those diseases, particularly for asthma, that asthma some of the causes of asthma must now be related to air pollution, and that asthma is not just a condition that's made worse by air pollution, but asthma may be caused by air pollution. I think there's increasing evidence. And this is really a summary of what I've already mentioned, through those mechanisms of inflammation, oxidative stress, and downstream of that. Now I want to talk about the other aspect of those components that can activate the epithelium and indeed cause the increase in permeability of the epithelium, and focus on the allergens. And first of all, I think one of the things that we were learning about allergens is that it can do that through the classical IgE receptor interaction, but also I'd like to start here as through non-IgE pathways, which has become a bit more evident over the recent years. So this is the way in which so these allergens, a lot of the allergens have protease activity, so it wouldn't be surprising to you if I said that the proteases can inactivate those a lot of those junctional proteins to degrade them and allow an increase in permeability, which will allow, in fact, those allergens to penetrate into the tissue. And also, by so doing, there's also an interaction with, for example, TRPV4 that leads then to the production of those alarmins, and also there's good evidence that those proteases work through those PAR receptors that then activates those pro-inflammatory cytokines that listed here, that has an effect on IFC2, and also they do have an effect in as I said, in producing those alarmins that also can activate ILC2, and then we have the downstream release of IL4, IL5 that has the a subsequent effect downstream. So these are the so and I've forgotten to mention the activation of TLR4 and MD2 through effect through the fibrinogen cleavage products through those protease allergens. So these are the non-Ig pathways that by which protease allergens interact with the epithelium. And I've already mentioned the production of the alarmin IL-33, but there's also another mechanism by which those allergens will activate the maturation of IL-33, and this is through this riboptosome IL-33 pathway that involves this pro-caspase 8 activation that has in effect in cutting this full-length IL-33 in the nucleus into the shorter version of the IL-33, which is in fact the activated form of IL-33. So I'll come to the IL-33 a bit later. So this is just some evidence. I've already mentioned the hydrocarbon receptor activation, but there is now some evidence that this AHR activation by pollution, diesel exhaust particles here, can be the basis for the production of those alarmins from the airway epithelium, in this case of severe allergic asthma. So, and this is as targets for treatment of asthma and COPD, and indeed we do have an anti-TSLP antibody that's now in use in treating severe asthma, and this is tezepelumab. And the antibody to IL-33 or to the receptor of IL-33, the ST2 antibody, are also now being. This is a study in our collaboration with Professor Kuo in T2 high severe asthma and the non-T2 allergic severe asthma compared to the other two groups. And right, okay, thanks. And what they've shown is that if you expose those epithelial cells to free IgE, you get you get a derangement of, and this is through gefitinib, a EGFR tyrosine kinase inhibitor. They also showed that through cross-linking, then there's a production of those alarmins, IL-33, IL-25, and TSLP, as shown here, both from mRNA and protein level in the cells, and also the amount of protein that's produced by the cells, measured by ELISA. So, so in severe asthma, there's this increase in this IgE receptor, high affinity IgE receptor in the epithelium of severe asthma, and the presumption is that the free IgE that exists in the surface epithelial liquid can interact with those high affinity IgE receptors, and that would cause a disruption of the epithelial barrier. And at the same time, the cross-linking of the IgE will, through the activation of the EGFR tyrosine kinase, will lead to the production of alarmins. So that really leads me to the fact that we do have those treatments for T2 high asthma, which is summarized here on this slide. We have, for a while now, the downstream effect targeting the downstream cytokines like IL-5, IL-4/13, and the IgE. But now we have the more important upstream target of TSLP, and hopefully later with targeting IL-33 or the IL-33 receptor or the ST2. And these have really been, for those of you who do treat patients with severe asthma, these have really been a very important advance in the treatment of asthma, with a lot of patients that would not otherwise have been controlled. There's really been a very, very good response to those treatments. I just want to finally finish on this, on the possibility that this increase in permeability is a reason by which we do get bacterial dysbiosis in people with asthma and people with severe asthma. So this is a study from our the cohort called UBioPred, which is a European cohort that we did about 10 years ago. But the analysis, the metagenomic analysis of the sputum microbiome shows that in the neutrophilic asthmatics, defined by having high levels of neutrophils in the sputum, is very distinct from the other eosinophilic groups, or the mix or the pulmonary group, and from healthy controls, by having very high levels of Haemophilus influenza and Moraxella catarrhalis. And in a mixed group, which is a mixed eosinophilic and neutrophilic inflammation, there was also a very high level of Haemophilus influenza. And you can see it was in the mixed group, and to some extent in the neutrophilic group, that this alpha diversity is lower, which means that there's less diversity of the bacterial species, which is not surprising, since a lot of the bacteria is composed of those two major bacteria that we know are pathogenic in the airways. And the question is whether this could be due to the fact that there's this increase in permeability that I've demonstrated is present in patients with asthma. And as this cartoon shows, that then the bacteria is quite free to enter the subepithelium and cause various activation. And we know that they can activate this inflammation, the T2 type inflammation, and as well as the non-T2 type inflammation. So I'd like to finish here, and the points to take away are quite straightforward. And obviously, the airway epithelium is a site of response to all inhalable insults that include pollutants, allergens, infection agents, and irritants. And the response of the airway epithelium to those insults, I think primarily causing an increase in permeability of the airway epithelium, a change in the epithelial surface composition of goblet cells, ciliary dysfunction, and mucus production, the activation of inflammatory and immune pathways such as type 1 and type 2, the interference with the production of alarmins and defensins. I haven't been able to talk about the defensins. The activation of the oxidative stress pathways, downstream consequences. And obviously, the response of the airway epithelium will depend on the type of insult that it is. And this is something that we're working on. And asthmatics are more prone to these effects, but I think it's important that I think we're getting evidence now that these insults may be the basis for asthma, may contribute to the formation or to the cause of asthma, and possibly of other airway conditions. And I mentioned how blocking the downstream consequences of those insults has been successful therapeutically in the treatment of patients with severe asthma. So thank you very much for your attention.